I've got a record.
That was your name, actually. Already.
Okay, so the first point I would like to make is that my English is not my native language, so I will probably speak very slowly, and I will probably understand half of your jokes. So if I don't smile, it's just. So after the idea was with Wax was to after he has presented some of the technical issues, some of the limitation, and so on. I was just giving a very short presentation about what I'm researching about the DIY community, DIY bio community, and this is both parts of my interest and both parts of my research. So I have a background in biology, and I'm shifting to sociology of science, and I'm interested in keeping my biology up, and at the same time understanding the historical and socio-economic condition for this. Type of culture to emerge. And so the idea is just that I go through different person, because this is just, the community is so small that it's very personal. It's very, if you follow some person, you will have like a kind of very good portrait of what is going on. And so I just go through a list of person presenting who they are, where they come from, and what they are trying to achieve. And so the next step of my research will actually go and meet this person, interview, visit a lab, and know what they are doing. And what is a little bit frustrating at this step is some of the specificity of the protocols or the results that I have obtained. They are, of course, not on the web. So you see some things, which is mostly media coverage of their activity, but it's very difficult to actually reach the information about the results of the experiments. So this is what I'm going to search with my research. And by the way, I'm this very awkward person that asks for interviews at the Hackspace, and I have no answer. And because I have to do some pilot interview before going there and mess up for my interview, because I never did interviews. So if some people are not afraid, I'm still searching for guinea pigs for my pilot interview. But I think it was just a little bit awkward to just send an email to the general group. I was like, hello. So, okay, the first person is, she's Meredith Patterson. I don't know if some of you maybe have already heard her name. She's involved with a lot of open source software development and a lot of hacking conference organization, and she's actually in Belgium. So she came to biohacking in 5 years ago, and in the picture you see her in the kitchen trying to do some experiments. And so again, I don't have any information about the results of her experiments. I just have information from media reporting her interview on what she's doing. And her project is creating a biodetector for melamine. So a biodetector is a living organism that is used to detect a chemical compounds generally. So how you can do this? You take your organism and you use the genetic material that it contains to connect the molecule you want to detect and the gene of the organism. And usually what you do is that you have a rapport system in which there is a GFP protein. So if the organism metabolizes or detects the chemical, then it will produce a fluorescent protein that you can detect. So this is called a biodetector. And research on biodetector has been going on for the past 10 years in order to mainly detect pollutants in the environment, because it's much more cheap than doing chemical analysis, and it's also a potential technology for a developing country to monitorize pollution in a cheaper way. So she was interested in melamine because I don't know if this molecule sounds familiar to you. It was like a toxic compound that was introduced in milk in China in 2006 and was a huge scandal about dietary products that were contaminated, and actually more than 10 children died and a lot of people were contaminated. So she interesting.
Was the contamination, was it that it was a chemical that was deliberately introduced?
Yes, it was a chemical that is used for producing thermostatic plastics.
Yes. And
has been, so there were industry that were selling powders contaminated on purpose with this powder, because they were producing a large amount of this powder and they were not able to give it on the market
Oh,
So they were selling it,
doctoring it with.
yeah,
They were cutting it
as
with
a protein,
melamine.
so if you added this to the milk, you will have a better milk.
Okay,
So,
I thought it was a food additive, but I wasn't sure whether it was an artifact of the processing
No,
or whether it was a deliberate insertion.
It was deliberate because they want to sell it out somehow, so they transform it into something you can add to a milk.
Was this something that it faked protein tests? Because the things would be tested to see how much protein they contained, and the value of the ingredient to some of these things would be based on how much protein it had. And the melamine would give a positive on that test.
I don't know this.
Okay.
I just know that they were selling this powder without telling the ingredients to the farmers. They were they were in the milk chain,
Okay.
and they were selling it as as a powder that will improve the quality of the milk in terms of like fat absorbance and this kind of stuff. But I don't know if there was in purpose of taking some analysis about the milk. And so you can see basically on the table what she's doing, and in terms of like what Max has been presenting, the techniques that she's using is cloning. So she's working with E. coli, which is this model organism for laboratory work. So she needed to get the sequence, insert the sequence into a plasmid, select as Bugs has shown, and then test it in the presence of melamine. But unfortunately, I don't have any data on the research, which that's why
she doesn't publish?
No, well, she published a lot of open source software, or mainly a lot of what she's done, and in terms of like code is published on the blog. But in terms of her research, nothing is published.
Is that a deliberate? Does she work alone?
I don't know.
Does she work alone?
She works with another figure I will present after, and she's. I think she's now. I mean, I've been following messages on DIY Bio London, in which they report that she's interested in opening a biohack space in Leuven, in Belgium. And she has found out a couple of other people. So this is really going on now. So I don't know. The next person is Tito Jankowski, and the other one I always forget because it's not a normal one. They are, by the way, Meredith, she comes from California, San Francisco, and I think it's very important to understand the social context where these people come in terms of the possibility they have to get access to this technology and to get access to these materials. So Tito and Wang are both students in biology. They have a background in biology and genetic engineering, and they've been, I don't know if some of you are familiar, have been educated in synthetic biology, which is a brand new science that tries to converge in engineering and biology to maximize the standardization of biology in terms of facilitating the design and fabrication of genetic modified organisms in very two words. And so they are in the garage of Tito House, and what they have in their hands is a gel box. So one of the gel box that Bugs has been talking about before. So it's just a place where you can migrate your DNA into a agarose gel to separate fragments according to the electric gradient and size gradient. And what is interesting is that this kind of machine, which are very at the basis of a laboratory equipment, you can find them secondhand or you can buy brand new. And this one is the cheapest one that is on the market right now, is $79 if you assemble yourself, and it's $199 if it's get assembled. And what is the another interesting part of this this gel box is the procedure for constructing it and assembling it is available on the website. So it's a fully open source hardware for biology. This is another project. Tito has been involved with another colleague of this is the OpenPCR project. So the thermocycler that you have been seeing in Bag's presentation are also available second-hand or new. Second-hand, you cannot spend less than $300, and usually you have a very old and unreliable machine. So they basically have tried to both to have like a cheaper, but also this idea of everybody being able to construct from scratch a machine. They have started this project, and what is interesting is that to kind of collect the money to do their own research and design, they have used a website called Kickstarter, in which they have advertised an auction. How do you pronounce this in English?
It's an option.
Auction, yeah. and they needed $6,000, and they have raised $20,000 for this project. And this is in the Bay Area, so this shows how much investment and how much different culture is going on in this area in terms of like biotech innovation culture. And so a little bit about right now, which is this one. They are circulating this prototype in different conferences like Make Fairs and these kind of places to present the prototype, to present their own activities. And they are writing on the website, on which you cannot find yet the entire plan of the open source PCR, that probably will be available in 1 year. So it's not yet available. But if you, I guess they are very easy to be contacted and to be engaged with. And basically, the electronic components are Arduino components. And then I assume the structure is laser cut and make-both components. So this is another person who was a lab in a closet. Her name is K.O. She's from Boston, and she has a background in biology. She also has been very much involved with the synthetic biology community in Boston that started at MIT almost 10 years ago. And she's been mainly working on second-hand material. So all the things that you see, like the centrifuge, the incubator, the thermocycler, have been all bought on eBay or other second-hand websites. And what is interesting of her project is the first person that has done DIY genetic testing. So genetic testing is basically when you extract DNA from an organism, and then you want to test for the presence of a gene or for the absence of a gene, and then correlate this presence or absence with a disease. So the interest is connecting a disease with a genetic marker that you can trace. And I never forget, I always forget the name of the disease. So her father has basically been, was found out that her father has hemochromatosis, which is it creates an excess of iron in your blood. And she wanted to find out if she was also carrying the mutation, because it's a genetic disease that you can carry out through generation. And she set up all the protocols to do the testing herself. So finding out the sequence, you need the primers to isolate the gene, and then testing the presence on yourself. So I find this is a very interesting project.
Did you do sequencing, or did you actually set up a sequencer?
No, she just did like a digestion at the end for presence absence of the gene. And I think in terms of like having a fully, fully circular project in terms of like machine aim and results, she's probably one of the person that in a very specific and small scale has achieved a fully effective biohacking project. So this is the dramaturge, and also, well, I think this you guys know much better this community about make-both and about how people are trying to use machine that reproduce themselves in an open source space. And I heard that recently Qatar is trying to organize like a biohacking space in Cork or in the region, but I don't know where is he or in terms of like he found out all the people at place or this kind of stuff.
He's got a Hackspace that started there in Cork.
Okay.
In Cork. But the bio one's a bit. I think that's basically him.
Okay, yeah.
But he's got a Hackspace at least. So he's got some style there. He's doing fairly well. My impression is he really knows his stuff.
Oh, yeah,
he really does.
Yeah.
He's a very smart guy.
I've been very impressed by the list of reagents and stuff that you have given to bugs. In terms of hardware, one other project is the reverse engineering of a webcam. So there have been a lot of people doing this, mainly artists involved with the Acteria network, which is a open source lab equipment for bioart, which is run in Switzerland in collaboration between Switzerland and India. And one of the founder of this project has first hacked a webcam by reversing the lens and transforming the webcam into a microscope.
Cool.
And this has been carried out in many places. One is Fruit in Boston, which is a knowledge hackspace type of in which biohackers or orbitates around. So yes, this is a hardware example. Then some other very important actors in the community is the DIY bio community that has been started in Boston by mainly two persons, Mackenzie Cowell and Jason Gold. And Mackenzie is a student in biology and very involved in the beginning of the synthetic biology community and the IGM, the International Genetic Engineer Machine Competition. And they started this network in 2006, if I don't mistake. And they have like they have different. The network has different aims. One is to promote the community and to negotiate with governments and all the security stuff in order not to be tagged as bioterrorism, which in America is a huge concern.
Well, that is essentially the discussion we had earlier.
Yeah, but in the States, the government was much more intrusive. So as soon as these people started to speak about setting up a lab, they had like FBI agents running around in place. So you see, like there are levels of suspicion from governments, and so they play a very active role in promoting, in explaining, in speaking about biohacking, and they have like two main projects. One is the one of Jason Gold. By the way, he's a very interesting person because he's like collaborating with George Church, which is one of the gods of DNA sequencing at Harvard University, and is responsible for the Personal Genome Project, which is the only place, is a database where like 10 participants has decided to get the entire DNA secrets accessible for medical studies. And his project in collaboration with these people, George Church and other people at the Personal Genome Project, is called BioWeatherMap. So the idea is that you create events in which people can scratch and collect bacteria from different places in a city, and then you outsource the sequencing because right now there is no hackerspace or biohacking space that has a sequencer. This is extremely expensive and is extremely slow, and I think this is slow and expensive. So they collect bacteria all around. They create events in which people can collect bacteria all around. They sequence the different samples that they've been collecting, and then they draw maps on bacterial diversity, and bacterial diversity changes in time. So this is for them like a kind of a cool project in order to use this technology to explore your environment into like a biomicrobial perspective.
Really awesome.
Yeah.
When you're saying DNA sequencing, so it's beyond any sort of like Hackspace or anything like that. What equipment's needed to actually do that? If you actually wanted to say do DNA sequencing, how much? I mean, is it possible? Is it realistically possible to build the gear itself, or design an open source variant, or is it just too complicated?
I think it depends on the techniques that you want to use, because some of them. Well, I've been only using sequencers, and I've never been, because it's a proprietary object, you never just like go at 8 o'clock in the lab and just like dismantle the stuff. And then, it's like, with the knowledge I have, very probably I will never be able to make it better for the next day when it's used by the other staff.
But I mean, it's the tech. I mean, does anyone know about how they work?
Yes.
Yeah,
You do know.
But, well, it's widely known. I mean, the techniques. The mechanism underlying how it works is very well established. It's very famous. Ask any biologist to be able to tell you. The specific machines themselves, the refinements, are more complex and harder. So you could, if you wanted to, sure, you could build a sequencing setup, no problem, but it would be extraordinarily labour intensive.
And this would take
very, very
years
slow.
to optimize like the reaction, the concentration, the timing, because it's like microfluidics. So you have to manage microfluidics plus detection. Usually you use lasers, so you have laser microfluidics.
So I mean, there are simpler ways to do it. You can do it with agarose gels if you have the rest of your life to do it. You can have just one lane in agarose gel for each base along the sequence. So you could have sort of 1,000 lanes for a kilobase, which is a very small gene, or small segment to do. Then you need to do that sort of 2 or 3 times, because some of the times it doesn't work and you need to get consistent results. If you really needed to, it's life and death, you could do it, but you'd be there for years, probably.
So realistically, I mean, it's too complicated to say, enough engineers throw at it.
When you can pay £6 or £8 to give it to university and they'll post you back a sequence, you don't want to be spending months of your time.
No, I was just curious to see if the technology you were talking about seems like it was too complicated.
Is it the sort of thing that would benefit from sort of distributed networking? So you're saying about agrogel, and you sort of, you've got 1,000 of them and you've got to sit there and watch them for sort of 4 days. Is it the sort of thing that if there was a network throughout the biohacking world, which admittedly at the moment is quite limited, but you sit there and you go, right, I want this sequence, you send it out, and everyone gets a sample, or a fragment of a sample, and
absolutely, yeah.
I mean, is that
or you could spend £10 and have someone do it for you.
Yeah.
Is it just literally £10?
It's very much easier to outsource this to a company that will do it for like literally 10, 12 hours.
It's labour versus cost and all that stuff.
It is becoming a lot cheaper. And there are systems, chip sequences coming out very shortly. And basically, if you're going to build your own, it's not what to do. Because eventually you'll be able to buy this.
Well, I mean, it's what I was wondering.
If you process it, but you
Yeah.
wouldn't want to.
Well, I've read about the chip processing and stuff. It's basically easier to buy those, which are going to be, incredibly cheap to do that rather than build one in the first place. But it's just interesting.
It would be a fun project. I've always slightly wanted to do it.
That definitely would be a fun project to get an old one in which the technology is a bit more. You can see stuff and understand how it works and have like a microfluidic.
How much do you buy like a second-hand one on eBay or something?
Sequencer, I think you cannot get a run for less than $300,000.
So how much do they go for new then? How much do they cost new? Like for brand new?
Depends on the model, but like for instance in the university where I come from, there's been a huge discussion among different departments to buy one, and it was up to 2 to 3 million of pounds.
That range of kit now,
exactly, and then
it's almost custom for your university.
So
the lab that did a lot of the human DNA stuff now, all that So I mean,
it's not only the costs outright.
It's going to be redundant.
That's what it's one of those things.
So if they could be good, we might get one.
It depends. Can you pick it up quick enough before the rate of everyone else buying it? Because I mean, I imagine it's still at the point where lots of universities will go, well, we'll buy a second-hand genome sequencer or even a third-hand one. I suppose it's waiting for that. For it to get to the point where the university will go, there's no point buying second-hand. At which point it may be cheaper to buy on-chip systems.
And there's been like a growing market in second-hand lab equipment because of like many biotech companies rushing with this crisis.
Before it gets out.
So the next project is No, it's cracking.
Gosh.
Oh no. Sorry.
No, again. Well, I don't know if the picture was really. No. Well, let's finish like this. The second, the other project of the DIY bio colleagues of Jason Bob Mackenzie Powell, he's an interesting figure because he basically got his parents buying a container, like a shipping container with a lab built inside. So this was like a shipping container for like mobile labs when you do analysis on like ecology or this kind of stuff that you move around, or even medical analysis like in developing countries where you need to. And so he now has this lab that is called BOSS Lab. It's the Boston Open Source lab, I think, something like that. And within this lab, he has a project called Smart Lab, where with an informatician is trying to wire up equipment and software in order to not record, to monitor the results that are used by some machines with a program in which you can see all the results when they're coming out from the machines. So this is another project in which informatics is a very relevant and very important type of knowledge in the community. And the last one, the last one is very recent. Is I think it's probably the only entire lab fully equipped that is existing right now. It's not existing yet, but it will exist soon. It's called Biocurious. It's an initiative. It's an initiative that's been taken by Tito Jankowski, the guy that you've seen before, and another person. And they have again put up requests for money on Kickstart, and they have collected $22,000 for starting the new lab. And they've really received the money. They finished the auction yesterday, so they will collect the money tomorrow. And so this is a completely new project that is starting. And I think
how much did you say it was for that? Sorry?
Biocurious.
Yeah. How much do you think they'd raise?
22,000?
Yeah, it's now 33,000.
Oh my god!
It was yesterday. I saw the same, but it's gone up overnight. It's gone to 33,000.
So who was I think the person who was giving more than 1,000 had a dinner with Carrie Moses.
Something like that. Yeah. It's crazy. It's like you pay. What do you get the most for it? You pay. Yeah, you get a special edition model, dinner with the people, blah blah, all that kind of crap. But it's currently up to $33,337 of a $30,000 goal. So
this is California, so it's like, it's a very completely different. I come from Switzerland. I can never imagine doing this in Switzerland. It's like what? So yes, I'm very curious about seeing the next step of this lab. So basically, this is what I call the serious people that have been really involved either in hardware development with open source, not idea but practices. And then there is a kind of a more vast community of people that are for instance, doing DNA extraction for strawberries at home, or there are people that have been. An interesting project that doesn't require a lot of technology was in New York. Was a group of girls collecting sushi from different bars and actually proving that the fish was not the one that was supposed to sell, and they did just by extracting the DNA, sequencing the DNA, and comparing the sequences with the species of from the MCBI bank. So this is a kind of interesting project. So, well, I guess this is a short portrait. I wanted to keep it short because I didn't know how much it would take the introduction. I think it was already a lot of information. So yeah, I think it's very nice to discuss now and ask questions if you have some.
How about people in London? Is there any mission?
Yeah, so London is they've been starting. So the first thing was I I think it's Marco Clark. Yeah, he's a student. He was a student at Imperial College in synthetic biology master, and he wanted to start something in London, and he actually created this Google group called DIY by London. And well, not a lot actually happened there. People started to try to meet up and never happened. And then the community was kind of hijacked by artists from the Royal College of Art, which started to do their own meeting, but then nothing really happened. And finally, the only person that were like sending messaging about I'm in London, what can I do? They've been directed here by Catal, and so this is what is going on in London.
I've been watching the group just sort of going, this has gone terribly. Yeah. It should have succeeded. There's enough people who are interested, there really are.
Yeah.
But it's been, as you said, it's been hijacked, and our group is terrible for that. But it's, I think there's enough interest to do some kind of like DIY bio thing in London. I mean,
so what would it take to make it work?
What to make it work here? People who have some vague knowledge. We've got two of those. I mean, the thing is, I wanted to do this. I came back from Maker Fair last year, and I met Catal, and he was like, "Yeah, you got to do it, you got to do it." And then people came down to London and gave us things to try, and but then this place kind of took off on its own, and it never really got taken off. But they're still really interested in helping us. So we've already got some people who are outsiders who want us to get going. Now we've got two biologists who know what they're talking about, supposedly. A lot of the gear is either we can make ourselves, some of it we can buy. We've already got some bits we bought already, and it just takes some vague knowledge to start teaching us these things. And I've learned a hell of a lot tonight. I don't know about anyone else.
I mean, when it comes to buying equipment, we already learned over the last couple of weeks that even buying equipment for thousands of pounds is realistic. Yeah, because we have a model.
It completely can be realistic. I mean you just need an end goal with it.
What would we need to do in terms of space? I mean, so I imagine some sort of bio lab would have to be a reasonably permanent fixture. I mean, I can't imagine it's the sort of thing you can sort of set up and take down in
the same thing as we've seen. Someone did it in their closet, and we've got a closet over there. It's got a fridge in it. I'm just saying.
Where would the beer go?
Well, somewhere else. But beer is important. Oh god, it's brewing. It's biology. It's feasible. No. Just bad images. I think it's completely feasible. I mean, if you guys wanted to sort of head something up with it, there's
what would be a good starting point? What is a good simple thing to start with?
My starting point is because I will leave for this field work, so I will try to take back as many information on the hardware, and probably try to get back like a prototype or like a model to do it and this kind of stuff. So my aim is to bring back this kind of basic hardware, cheap stuff. And then really depends on, I think it depends both on that. I see two kind of way to see it. One is like development of open source hardware, which is something that doesn't need per se a lab setting. Or the second one is actually carrying out experiments, depending on you want to do. Like for instance, you say you wanted to do an electrophoresis gel. This is enough of a motivation to make a list of things that are necessary to do an electrophoresis gel, and to try out like a funny experiment about extracting DNA from yourself and digesting it in some way. I don't know. It's really.
I think that the great example was the sushi example of like people like tracking down. That's brilliant. That's so brilliant. It hurts. Like that's the kind of thing here you go. We're going to go to every sushi restaurant in London and check if they're lying. Sign me up. That's a great project.
Just go to every sushi.
And,
it's the kind of thing.
It's a very.
I mean, I think that there's a great crossover there between people who brew beer here and things you can start doing with beer. Like you were saying, glowing beer.
I was going to say, how could that be to set up?
What, for brewing and that kind of thing?
Well, I mean, the actual brewing, because I've actually got cider brewing in bugs in my flat at the moment, but sort of bugs, how difficult would you reckon it would be to tag yeast with GFP?
I think it's safely for human consumption.
So safely.
Yeah. You can be the guinea pig.
If we decided we wanted to do it,
guinea pigs.
I know in the lab it'd be trivial to do. The thing I haven't done before is transfecting bacteria and plasmids into yeast.
I would imagine it's very easy to do, but I've never actually tried to do it. Because it's doing it with some kinds of bacteria. If you have access to the bacteria, you put the plasmids in the tube with the bacteria, you heat the bacteria to 42 degrees, the bacteria go, oh god, it's hot in here. And for some reason...
I'll have to take off all my clothes.
And for some reason, they take in the plasmids from the media they're in and start expressing them. The reason is well known, but I don't know. So... Or sometimes you need to use sort of slightly nasty chemicals to do it, but then you can get rid of the chemicals and then deal with the stuff very safely. I don't know how it's done in yeast. I assume it's a very similar to protocol. It's something I've done myself. It's only something I can look into.
I mean, because it's the sort of thing that I don't know about other people, but I would be very interested in having glow-in-the-dark beer.
Yeah, but would you consume something you've made in the lab without testing it on animals?
Because animal?
I test it on him.
Because I wouldn't consume it, and
personally, I wouldn't drink it until like a month after other people have been doing it. And even then, I'd hesitate.
So I don't think anything for human consumption is viable unless somebody wants it.
Just saying.
Yeah,
I
everything
agree.
in this
You're
now.
not going to consume anything that's you've made yourself.
And I don't want to test on mice.
No.
We could do something very similar to the sushi experiment, except we could go to Brick Lane and check that the lamb is actually lamb.
No, you don't want to do that.
No, I mean, it would
never
shatter our
outrun them
illusions
again.
and we'd never go there again, but that would be quite a good thing.
Yeah.
But is our first step just gathering all the equipment that you had listed on your slides?
Not all the equipment, enough to do a small
I think the thing is choose a project you want to do, and then think about
But
it.
the thing is,
there's going to be some feedback.
The stuff you can do, like extracting DNA from kiwis, things like that, which you can do just with stuff you've got in your house. It depends how complex you want to get. There's a lot of projects we can start off. So there's actually a really good site. I can't remember the name of the site now, but yeah, there's a really good site for lots of bio projects to sort of ramping up in complexity. But they stop at the point where you have to spend a couple of grand on a piece of equipment, which is now fairly standardised or open source or that kind of thing. So it's when you get to that point there's an issue, and I think that's where most people find it interesting. Because stuff you can do in your kitchen as a kid, I'm not a kid anymore. I want to do something that's more exciting than that. So I think we just need to decide exactly what we want to do or what we want to mutate, that kind of thing.
Being said, with regards to stuff in the kitchen, do you want to try making colour changing drinks?
Oh yeah. So we found
beer related. Can we go to different brewers and check that the meats that they're using from where they say it's sourced?
Yeah,
That would require going and drinking all that is, but
that's a terrible one.
Would they let us in?
I wouldn't imagine much DNA makes it intact in the beers.
Yeah.
Exactly. I mean, that's the same note as the sushi crawl.
Do you just want to propose a drunken sushi crawl? Just have the guts.
One consideration here,
Drunken on beer.
So within the last month or so, I've been. It's been hell, I tell you. I've been doing paperwork. I did science, I don't like paperwork. But I've been filling out my lab's applications for a license from the Home Office to work with GM organisms. And the fact that there are different levels of biohazard that you have to worry about. Most of the stuff we do in my lab is biohazard level 1, which is, try to avoid drinking too much of it, but you're not really worried. And so, given that my lab has had to apply for a license to do this stuff, I don't know where the home sort of biohacking stuff falls legally.
So my feeling on that is that here we have no insurance, and we kind of trust people aren't dicks.
Yeah.
So we should probably speak to your mum.
We possibly should, yes.
Yeah,
but
if you
a lot
let
of
them know,
things.
they can say no.
Yeah. Well, this is the thing. They can say no. So
But if they say no,
get
that's
there.
probably for good.
But it's just.
I suppose that comes
Do you
down
want this
to
piece
once
on
we've
the
decided-
save video, by the way?
That's fine.
I suppose the thing with that is it comes down to we need to decide what we're doing before we talk to the environment. I mean, if we're sitting here going, let's try and reassemble smallpox, I imagine the environment...
Don't say that on video.
Sorry, quit this. But I imagine the Environment Agency and possibly the MOD and the government in general might have an issue with that.
Yes.
But if we're saying, well, we want to try and make yeast that glows in the dark, we're probably not going to care too much unless we start trying to feed it to pumpkins.
Fluorescing bread would be awesome.
Well, it would be quite fun, but
Yeah.
you meant to do it on the slide.
We might notice.
Take it to Wetherspoons, they won't notice.
But it's glowing in the dark. No one wants. Yeah.
It'll take a while to get the more complicated stuff together, right?
Yeah.
So I would suggest a simpler project to entertain us and get some results straight away, while other stuff is kind of ongoing.
What I was trying to get to before is that I think there's a lot of things we can do to ramp up to get people to the same level, or at least understand exactly what they're doing. Where a lot of these projects they're set out so that you can go, do this and this, and then you've got so-and-so, and everyone goes, wow, but don't really understand what happened in the middle. Yeah. So it'd be great to have that explained by people who actually understand what the hell is going on, to get everyone to the rough same level, and then we start doing some interesting things. I don't know if that's interesting for you guys, seeing as you work on this professionally, and that sounds dull.
I quite like that, actually. Sorry.
Yeah, one of the projects, I've worked for a laboratory that does public engagement with people, and we're setting up using the synthetic biology BioBrick database and MIT, and we ship quite easily genetic cards. We did an experiment in which people were trained in 3 hours to genetically modify a bacteria to make it smell like banana. So this is a kind of a project.
You actually missed out BioBricks entirely from the presentation.
Yeah, because not all of the people that have been doing genetic engineering, Mary Patterson, she's not running on BioBricks. And most of the people are not yet to the stage in which they actually do genetic engineering. And if they do it, they are either already in a biotech company and they kind of do half of the work on the bench in the company, and then at home they do something, they continue the work. Either they are very much within the BioBrick community and then they are just, doing their PhD and then they're doing a little bit of their PhD at home. So
my point was really is that I know what BioBricks are.
Yeah.
I imagine most people don't know what BioBricks are. Who doesn't know what BioBricks are? Well, there you go.
Okay, so BioBricks is within this new discipline called synthetic biology, and with this purpose of standardizing biology, because there is a critique, which is a fairly superficial critique of this new discipline that has to justify itself as new and therefore cool and therefore raising money and so on. They have decided that biology is not at all standardized and they need to standardize it. And so they choose the analogy of electronic components, and when you want to be at the table, you want to be sure that that you can buy a surface, and this surface has a certain number of attributes, and that you can use this, you can know these attributes, and they are always the same. And so what they have been doing, this is part of the International Genetic Engineering Machine Project. So basically, they've been using students to develop these parts. So a part is a gene, which has a specific function that you use to do or whatever you want to do. And they ask people to study the gene in terms of which type of promoter, so how it is activated, in which situation it is activated, which is the context that it's optimized in order this gene to produce a protein, and so on, and put all this information in a database, which is available from the people of the community and is available on demand from exterior people by telling that you kind of run a lab or you are a part of an institution that's recognized as a loyal institution, this kind of stuff. So there is right now they have up to I don't know, it's 50,000 parts, so 15,000 components. There can be gene, there can be promoter, there can be stop sequences which stops the reading of the gene, and so on and so on. And so basically this database is mostly used by the student within this competition. They go and they browse in this database and they choose pieces. Then they made it synthesized by a company that synthesized DNA, or they try to extract it from another E. coli or another bacteria that has this gene. So this is.
The thing I read about it was that they were trying to get together something that was kind of software, really, where you said, I want something that does X in response to X, designing something specifically, and it was you take this and this and this and this. It's like Lego, and you go, and it makes something that does that for you. And they're trying to get fit. I mean, is that right interpretation?
That's maybe a level higher. But primarily it's sort of a modular system for building all of it.
Yeah, that's what I read the aim of the project was to get to. So they need to designate Or, I mean, you need to know what all the Lego bricks are before you can stick them together, and that's what they were trying to do at the moment. The end goal was to have something where you can design something that did X, and they would tell you exactly the steps to get there, much in the step of a Lego manual. But yeah, it's usually the same sort of thing.
The problem with biology is that you have a very specific function that is provided by a very complex connection of elements. So if you say, for instance, I want to make this bacteria that smells banana, you have to go back to the metabolism and the molecules and then trace which of these molecules are part of a genetic first compounds, which are part of a secondary metabolism that are produced by protein produces this molecule. So
it's more like an API.
So what you actually can optimize with or make in an automatic way with a computer is when you have your structure or your what we call it is a construct. So when you have your construction with the genes that you think that they are producing this product, you can test it, you can simulate and relate it to experimental data. If you for instance, optimize sequences or optimizing promoters and so on and so on. But as far as I know, I never see somebody going from function to the construct in a mathematic way through a software.
Yeah, I mean, I know it hasn't been done yet, but that was the aim.
Yeah. This is part of the synthetic biology.
So I guess one other way to look at this is you try to take a system that is not designed to be understood by humans and to be manipulated by humans and turn it into something that is and can be.
But it's really interesting. It's except of this database, which is used by a very small community. Synthetic biology has been mainly very often redoing what the genetic engineering has been doing for the last 25 years, with this very nice and challenging whatever argument that they're doing something new because it's engineering. It's not genetic engineering. So there's been
it's
also very fresh community and it's going through a lot of challenge and criticism about people that have been doing genetic engineering for 30 years.
It is very appealing though because there's arguably a lot of art as well as science to cloning, in the sense that it comes back to this idea that DNA isn't just information, it's also a physical object that has a structure. And the same is true, of course, for proteins. And so if you put these sequences together, there are some sequences that just don't like being cloned. And if you have two protein domains, you stick them together, they won't necessarily fold into the same shapes or have the same...
domain? It's just region.
Region, yeah. Two chunks of protein. If you stick them together, won't necessarily fold into the shapes that they used to, and they won't necessarily have the same functions that they used to. And there's a lot of fuzziness around that. So I think a big part of what BioBricks are trying to do, at least my understanding of it, is that they're trying to kind of rationalize this as far as possible. So they're trying to say, okay, this subunit, you can put it into any part of any sequence, and it will always have this function. How achievable that is, I don't know. But I think this is the idea, to take some of the art out of cloning and bring it closer to, as you say, closer to engineering.
Because what is interesting is when you browse the database, actually, it's very, because when you create the system, you have to ensure that everybody is reporting by using the same standard values. They characterize a promoter in the same way, if not the values are not comparable, and then you have the same problem biology has been facing for the last 30 years. And when you actually browse through the database, a lot of things are missing, are inaccurate, because it's students' work. And they have a very short time, and they are under a lot of pressure to get the project finished, so sometimes they just deliver something without actually checking everything. So now what they've been doing, they have created kind of I don't understand very well the economic or patenting system is around this, behind this structure, but it's a place where they basically are doing the same work in student but in a professional way. So you can call them and say, okay, I would like to have this construct, please optimize it for you, blah, 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 blah. So this is kind of a spin-up or whatever spin out of this database into an economic and professional type of culture or system.
How difficult is it obtaining the various bits of gene listed on the biobook? I mean, because that sounds like the sort of thing that I imagine we could do quite happily here is run experiments checking, does gene XYZ actually do what we expect it to do? Does gene XYZ do what we expect it to do when we find something else? I mean, are these are these DNA sequences easy to obtain? Do you have to sort of sign into the bio block? Do you have to sort of get some form of authorization? Is there sort of like a licensing system on it?
No, there's no. I mean, they've been in November of the last year. They've been issuing out like a proposal for an open source protection structure for this database, which is not very clear because it's a mixed system of some people can you can extract like a construct from the database and then you can either put it back without asking anything, either patenting and then constraining utilization of the people in the database. So it's a mixed model, which is not very clear now. It's not been implemented. It's still under discussion.
So at the moment, bits of it are.
Yeah, there is no clear license.
Yeah, so bits of it are sort of being patented happily by Monsanto and Co, and bits of it are being sort of left.
No, this is just so the parts that are in the database are not patented because as soon as it's illegal. But they've been trying to create a legal system in which people are pushed to put back parts while they've been working on it, and at the same time allowed to kind of have some patenting possibilities on the parts they've been working on.
It's amazing how similar this is to software.
Yeah. Well, interestingly, some of the people in this community have been very near, Tom Knight's one of the founders, has been like a colleague of Stallman back in the 70s of MIT Artificial Intelligence Lab, and they have these open source cultures definitely coming through software development. But for the question about how easy it is to access this, it's like my own experience is that I could ask these sequences because I was within a university. But I know that there are people that have access to these sequences without being in university.
And then you just get hold of the sequence and send it off to a company and go, I want plasmids with this sequence in it, and they send it back to you for some fee.
Or some of them, they just, what I've been asking is because definitely, if you ask, the option I had is either I get the sequence and I made it sensitized, which was $6,000 for a sequence of because it's $0.75 per base pair. So $6,000 means which is almost the same size in terms of DNA. And or you receive the bacteria with the plasmid inside, and then you extract it and you do whatever you want. So which is much cheaper.
Mm-hmm. Why is that so much cheaper?
Because you have it for free, while the other one you have to make it sensitized for it. Yeah, because they really, you receive a piece of paper in which you have a stabilized culture of bacteria, and then you just need to put this bacteria in a fresh media, and they made them grow and reselect them. And then
It's like the equivalent of paying for a highly optimized compiler.
No, it's the difference between buying the components and buying the finished product.
Yeah.
Yeah, so you're paying to...
I kind of get what you're saying. It's like you're buying the compiler or you're buying the finished product.
You're paying for the
things producing the product.
and the output. You can't be so sure that you're going to get the right results.
Yes.
So what's the easiest thing that we can do that would still be interesting? That we could be doing as well?
Testing some products on the market.
I think we should put all this on the wiki.
One resource that would definitely be interesting is just lists of websites, and I'm sure those lists exist with services. For example, sequencing services. That's a good starting point, right?
And also just nice if
electrophoresis.
Yeah, exactly.
It's got to happen.
Because, for instance, the electrophoresis is a step within an experiment, so it would be definitely be part of it. So.
Yeah, we should get that stuff fairly early on.
Actually, what would be interesting is where can you, what are good resources to research known experiments? To what are good catalogs of experiments you can do? Where do you even find out about the possibilities without being a researcher yourself?
Well, there are some of them on Instructable. This website
but are there like books of recipes?
Yes, but you have to belong to the network. But they put the recipes with the protocols. But what I've been seeing is that they are not very much into. They are much more like, let's say, non-living experiments doing. So the only one I've seen is like the Biohacking project was there. And yeah.
So really, it's the new frontier. There's nothing.
Well, there's a lot of experiments that have been done a lot. In terms of like having a platform.
It's very exciting because at the moment, there's basic infrastructure doesn't really exist. It exists in corporations.
What is this kit for? I think it was a kit where you can run gels and test the species of vegetables.
Really?
Yeah. So you have like a microelectrophoresis chamber. But basically one really easy thing is to test stuff. So you extract something and you look at the DNA and you tell you something about it. This is very easy. Like the sushi.
Is it the hello world of biohacking? Is that
well, and for me, I don't work in stuff, so probably.
Is that the simplest thing? Is that the first thing you do when you're trying to just like get a grip of what?
I think it's probably simpler than that.
Yeah, the simplest stuff is you could watch microbes on.
It's probably like culturing your own bacteria on gel. That's probably the simplest thing I would imagine. And then go.
I think the simplest would be like extracting DNA from your own things and then digesting it and running through a giant But
I guess we could start with that. Or is that too simple?
I think everyone's going to be interested no matter what happens.
Yeah.
Bacteria and shit. I mean,
next Tuesday we can do the thing extracting DNA from strawberries.
Yes.
Yeah, we should do that.
I think you need, what is it, it's like gin, washing up liquid and salt water or something.
I didn't even need gin, it's just washing up liquid.
I think we need gin.
It might be helpful to do a bit of an assessment of what skill sets we've got around and who can do what and who's interested.
Yeah,
I mean, I think the thing that's going to be most interesting for people, I mean, everyone likes sort of playing with the stuff and using it, but with regards to designing the open hardware and making that kind of thing, there's going to be a lot of interest in that. And then actually being able to use it is a bonus. So it would be great to be able to test it. So
I think it's also nice to imagine this like in a fairly slow time scale in which like maybe it takes like 3-4 months to develop for the open to just install or optimize or whatever, and then you have a PCR machine and then you go a step further, and while you construct the machine you understand stuff that enable you to think to a project. So
I mean, I think the thing is, if we want to run it here, the easiest thing to do is to have one night a month or more than one night a month where everyone who's interested just meets up and keeps coming. And you're going to get new people attending, so not everyone's going to be at the same level, but they can learn from people who've already learned, and you can keep building up. And there are going to be some people who are way ahead, but they can teach people who aren't as ahead. So if you're interested in that kind of thing, I just choose a night and start turning up.
Yeah, the only thing is because I've been in a way collecting information is just the next 2.5 months. Like a little bit of a pity to start this stuff now for me.
I imagine though that there's enough of this that's available online for us to be occupied for 2 months.
Yeah, what we can do is have a.
What, in London?
I'm sure we can find people who are interested in.
I meant kind of interested and with knowledge.
Yeah.
Again, in London, that's not going to be as far as you thought. There's people who've already shown interest who just, there's nothing going on, so they don't get involved. So we can contact them.
But I think it would be very interesting to see what people have as a project and what to start from in terms of very small steps.
So maybe tonight, make a page on the wiki, and then over time we can build up project ideas and suggestions and resources. People who are interested and get a discussion going on that. It's perhaps something we need to absorb some information and have a think about, because I'm really bad at thinking of projects like this because I'm so used to having the luxury of a fully funded lab. It's just really hard for me to think, oh yeah, I'll do that, it's trivial, except for that machine and that machine and that.
So to start with, think of something that you do in your lab, and then work out which bits are missing, and then
Cure
start
cancer.
challenging people. Not that, but start challenging people here to try and solve problems, and then you're not going to have the same people who are actually doing the experiments, but some people will spend days here and nights solving problems that you with this equipment don't have.
I mean, there are definitely bits of lab equipment that are very expensive to buy, but I'm pretty sure could be built mostly out of Lego and a hope.
Can we please build a Lego centrifuge?
Only if I'm far, far away.
Shouldn't have said that.
No, it's with duct tape.
No, not Lego.
You say Meccano or connect easy, but Lego.
Seems like a logical conclusion though. Should we call the camera so no one's feeling a bit self-conscious anymore?
Should we disperse?
Bye!
Thanks a lot.